Hello, viewers. Welcome to another episode of Research Digest podcast. In this episode, we'll talk about infectious diseases and, in most part, about COVID 19. So, today we have a guest who has extensive experience on COVID 19 research since 2020, and he has a lot of wonderful publications. Let me introduce you with our guest today. We have with me Dr. Mohammad Rubayet Hasan, Tanvi. Dr. Hassan is currently working as a clinical microbiologist in Hamilton Regional Laboratory Medicine Program. He's also an assistant professor in McMaster University, Canada, in the Department of Pathology and Molecular Medicine. Tanvi bhai, welcome to my podcast.
Thank you so much, Banna, for a nice introduction. I'm happy to be in your podcast today.
Yeah, my pleasure. So let's start our research talk first, and then we'll move on to your career part. So you have published a lot of papers. You have over 8,000 citations, and you co-authored several publications in very high impact journals like Lancet Infectious Diseases, Nature Communication, Nature Medicine, Vaccine, and many other journals like that. So uh, let's focus first on COVID-19. So are we now out of danger from SARS-CoV-2? What do you think?
Yeah, that is a very good question. Well, as a scientist, I would never rule out, 100% possibility of an oncoming threat, right? We never know what can happen in future, but based on our knowledge, right, of the pandemic, starting from the pandemic as of now. So the reason why we are not so worried about COVID-19 now is the herd immunity, right?
Yeah.
So, and in the first hand, at the beginning, why COVID was a problem. SARS-CoV-2. I mean, I think I've been telling this for more than 5 years now. SARS-CoV-2 is actually not a particularly very dangerous virus compared to many other viruses, right? So
Okay.
if you compare with even the SARS-CoV-2's other sister like MERS, Middle East coronavirus,
Yeah.
which had a way higher mortality rate than the SARS-CoV-2, right? So,
Yeah.
but why SARS-CoV-2 was a big problem was because, first of all, it's a new virus, and the population, is not immune to that virus.
Mm-hmm.
So we are susceptible to that virus. Number one, and then despite mortality is low, the other properties of the virus that made it so dangerous is its the transmissibility or infectivity.
Okay.
So the virus is like, I think its biggest potential is that it could easily transmit from one patient to other patient,
Mm-hmm.
and so the infectiousness in other ways, right? So
Yeah.
again, remember, SARS-CoV, even if you count infectiousness, SARS-CoV-2 is not the virus that has the highest infectiousness. If you compare with measles, say, for example,
Okay.
or varicella, like the one that caused chickenpox,
Mm-hmm.
they are lot more infectious than SARS-CoV-2.
What
But
about
measles or
Ebola? How do you
chickenpox,
compare with Ebola?
sorry.
How do you compare with Ebola? Isn't that more
No,
dangerous?
Ebola is infectious, but the route of transmission. Now that's another factor, right? So route of transmission is also a factor. So the viruses that can transmit through air or droplet, they are
Mm-hmm.
more dangerous because they can be easily
Right.
transmitted from
Yeah.
one patient. So Ebola is more of like close contact.
Okay.
So that's why it's a very dangerous virus, but in terms of infectiousness or transmissibility,
Mhm.
it is inferior to SARS-CoV-2. But SARS-CoV-2 again inferior to like measles. But measles is not a problem for us. It was a big problem. It's not a problem for us because of the widespread
Mhm.
vaccination, right? So that was the thing, SARS-CoV-2. So we have seen like how SARS-CoV-2 kind of paralyzed the entire world.
Mm-hmm.
We learned about the virus. We learned about how the virus transmits from one patient to other patient, and because of that, we implemented all these social restrictions, right?
Yeah.
Non-pharmaceutical interventions. So because of that, so now the reason we are not so worried now, there are a few factors, few reasons for that. Number one is the herd immunity.
Yeah.
Because of the vaccination as well as natural infection, I think the majority of the world population are now immune to SARS-CoV-2, right? And what
Mm-hmm.
happens in herd immunity is that, even if somebody has COVID-19, that person is guarded by like, four or five other people around. They are immune to the virus. So even one person is susceptible to the virus, he or she is not able to transmit the virus because
Yeah.
the others in the herd are working as a wall, right?
Yeah.
So
So it's not spreading like
exactly,
before.
exactly.
Yeah.
And the other reason is, the viral evolution or the mutations, right?
Okay.
So as you have seen that how many SARS-CoV-2 variants emerged over time, right?
There
And
are
we have
many
seen
varieties
that
of.
gradually, we have seen the parental strain, and then eventually one after
Mm.
one, alpha, beta, delta, omicron, right?
Yeah.
And then we have seen that certain variants can cause more severe disease than the other, right? So they have all different kind of mutations happen in the virus to increase its fitness. It's called
Mm.
viral fitness, right?
Yeah.
So eventually, there is a theory actually behind it. It's called convergent viral evolution theory. That means the virus would keep on mutating, right? And normally,
Mm.
even within a host, when a virus mutates, right? So only the mutant that provides some, fitness advantage or survival advantage, right,
Yeah.
would sustain and would transmit to another one. So, and that is also linked to the mechanism how the virus infects the human cell, what sort of receptors they use, what sort of biochemical reaction happens there. So now we think that, so much mutation happened, right? So
Yeah.
eventually the all the mutations, converged in one type of variant, like an Omicron variant. There are a lot of lineages and sublineages
Mhm.
of Omicron, but WHO, and also many other authorities, especially in the developed world, right, they keep track of the variants, which variant is emerging now, and whether this variant is any
Okay.
concern in terms of whether this variant is able to cause more severe infection, or is it able to evade host immunity. We right now we have pre-existing immunity
Mm-hmm.
against almost most of the variants that has emerged so far. So the new variants, it's not that the new variants are not coming. Every day there are new variants are coming, but we are not too concerned about that because there is no information yet to us that this variant is capable of doing an extra severe disease, or it limits the vaccine effectiveness, right? So
Yeah.
that's why if you go to the WHO website, you will see that right now there is no variant of concern. So
Mm-hmm.
there is variant under monitoring, there is variant of interest. And the last variant of interest was the JN1 variant of Omicron, which was actually in 2023. So that means in the last 2 years, we did not have any variant that is of any particular concern.
Okay.
And if you
So,
ask me,
uh,
so WHO tracking or developed country tracking are limited to their countries, what's happening in the developing world, right? Where there is no routine surveillance. I would say,
mm,
even after that, even if there is no surveillance, I'm sure that if there is a major variant that is causing a significant problem, we would have learned it by this time because of epidemiological reason, right?
yeah,
So,
yeah.
yeah,
Yeah, world is connected and it would definitely be noticed.
exactly. Yeah.
Uh, so here, I saw different pharmaceuticals are updating their vaccines every year, then do you think this is really important to maintain the herd immunity in the society to get vaccinated every year? Or do you think even if we don't get the vaccines, it should be same?
Yeah, no, no, the vaccine should definitely be updated based on the, I mean, this is not new actually. This is not only for COVID. If you look
Okay.
at influenza virus, there is
Hmm.
influenza vaccine. So
Yeah.
influenza vaccine is also updated every year,
Yeah. Flu vaccines are common.
depending on the circulating strain, right? So,
Yeah.
so lucky that we have mRNA vaccine now, which is actually way easier to develop and quickly develop. So yeah, so I think the latest vaccine, recommended vaccine, is the 2024-2025 version of the vaccine. Actually, only three companies right now provide the vaccine: Moderna, Pfizer, and Novavax. They are continuously updating this vaccine. Yes, the recommendation is definitely to take the most updated vaccine, because
Hmm.
the if we use the earlier versions, they may not be as effective. Especially Omicron, brought a lot of change in the antigenicity. So
Okay.
there was a compelling evidence actually
Hmm.
about that. So that's why it was necessary to update the vaccine. But in terms of recommendation about to maintain the herd immunity, I mean, it's not that COVID infection is not happening now. We still, because we collect data every week.
Yeah.
How many COVID-19? Like right now, we had it has become now a, like, seasonal virus, like other seasonal virus influenza. So
Hmm.
like now winter is approaching. Right now, we actually already seeing the higher
Hmm.
number of COVID-19 positive cases, but
Okay.
it is still not a problem. But it could be problem in patients who has comorbidities and who
Hmm.
are vulnerable group, right? So it is a really good idea, especially people, especially all elderly people, people with cardiovascular disease, people with kidney issues, people with diabetes. They should, at least on an yearly basis, they should take the vaccine, same as the influenza vaccine, because
Okay.
the risk of severe disease in those patients is much higher. And it's not that death is not happening because of COVID anymore. There are still death happening in the
Hmm.
hospitals, and we don't panic about it now because the rate has gone way down. That's why.
Okay. Okay. Yeah, that's an important information. Thanks for sharing. So I have a question on the virology perspective. So
Mhm.
uh, do we know that why some viruses tend to mutate a lot more than others, or uh, some make more variants compared to others? Why does that happen?
Yeah, it's an interesting question, and it is actually particularly happens more with the RNA viruses, because it
Hmm.
happens because of the error they make. Like, for RNA viruses,
When they copy their genome.
exactly. So for RNA viruses, there are more step, right? In
Mhm.
DNA viruses, the replication is directly, right?
Yeah,
When
yeah.
it replicates from DNA to DNA, right? So for RNA viruses, you have RNA dependent RNA polymerase, so which is called RDRP, so which needs to make a copy DNA first, and then the copy DNA would transcribe back again. So so that's why there are chances of base pair error is much higher with RNA viruses. And the fact that some viruses are capable are more prone to mutation is because of that they lack the proofreading enzyme. So some
Hmm.
viruses has some proofreading enzyme, and
Okay.
some viruses doesn't have the proofreading enzyme, or their machineries are not well equipped to correct the error. So that's why some viruses are particularly more
Okay.
prone to mutation. Yeah.
Okay. Yeah. That's really
The other thing,
interesting. That.
other factor is very important, is that the scale of replication is also important. So in case of COVID-19, we have seen it so much. The only reason is there are millions of cases, right? So the virus is getting more and more opportunity to replicate. So the chances of mutation, although the rate of mutation per virus is not particularly high in SARS-CoV-2, but because of the massive scale of transmission, right?
Mhm.
And the mutations of those variants become a bigger factor.
Okay. Yeah, yeah. Uh, I was also checking some epidemiological data a few months back. So I noticed RNA viruses are responsible for most of the worldwide viral pandemics that happened in last 50 years. So that's really interesting information that you shared. So like COVID-19, HIV, MERS, all are RNA viruses.
Yeah,
Well,
so not necessarily all viruses has the potential to cause pandemic. So
mhm.
this is also another property of the viruses, right? So there are
Yeah.
many factors there, and
Mhm.
one of them is high transmissibility, and the
Yeah.
other is that route of transmission. The viruses, that transmit through like blood-borne pathogens, say for example, right? So herpes viruses, say for example.
Mhm.
So they're they're way much less likely to be transmitted because they need to get into the blood to cause into infection. So that's why
Mhm.
they happen through needle stick injury or through other means. So the biggest problem are the viruses, that can be transmitted by air. So because it is very difficult to control that.
Okay. Yeah. All right, thank you. So, uh, let's talk a little more about infectious diseases. And you have experience on working mostly some infectious diseases spreading in South Asia, Bangladesh, India, and those regions. So, what are some of the emerging infections that people should be care about?
Well, there are, both short term and long term implications for certain infectious diseases. So if you tell me about longer term, I think the biggest problem right now in Southeast Asia is antibiotic resistance.
Mm,
So, because of irrational and uncontrolled use of antibiotics in our countries, right? As well
yeah.
as no infection control measures, neither in the hospitals, and lack of hygiene. And so all these contributing um, to the emergence of some of the extremely dangerous superbugs.
Okay.
And unfortunately, I mean, there's so many things happening behind the scene. We don't
Mm.
even know. The reason is there's no surveillance. There's no screening testing done on antibiotic resistance in Bangladesh, right?
So
So, but
for
I'm pretty much sure, like even if you say like 10 years old data, I know that in PG, say for example,
Mhm.
in PG hospital, in ICUs, the rate of carbapenemase resistant superbugs were like almost 25%, which is like less than 1% in developed countries now. I'm talking about 10 years ago. And
Okay.
imagine where it has gone right now. In a
Mm.
recent paper, I think about, I think last year, there was a published paper from ICDDRB, Bangladesh,
Okay.
which showed, the rate of colonization.
Mm.
You know what is colonization? Colonization means, they will take rectal swab, and then they will test for antibiotic resistance bacteria
Right.
in the
Yeah.
rectal swabs, right? So testing rectal swab means not necessarily means the patient is infected. So if you find a antibiotic resistant bug in the rectal swab or armpit like axilla or nair, right? So if you find them, that means they are carrier. They
Mm.
are carrying, they are colonized with these highly resistant, drug resistant bacteria, and today they are not causing a problem. Tomorrow they will become a part of an infection in the same patient or a different patient, and then it will be extremely difficult to control that kind of infection, because nowadays there are a lot of pan drug resistant bacteria, and I'm
Mm.
sure there's a lot in Bangladesh circulating now, for which we don't have any tool, any antibiotic. So
Yeah.
all antibiotic, available antibiotic, even the biggest guns, like
Mm.
highly broad spectrum antibiotics has become ineffective in those scenarios. So I'm sure in the background, a lot of people are dying in Bangladesh, especially the susceptible population, like the patients who are cancer patients, say, for example,
Mm-hmm.
they are receiving chemotherapy, right?
Yeah.
So the chemotherapy, and they're also receiving immunosuppressant. So their normal immune system doesn't work against bacteria.
Yeah.
So for them, when there is an infection, the only tool is antibiotic. And if the antibiotic doesn't work, there's no way to keep those patients alive. So even before cancer kill them, the infections are actually killing them. So
Yeah.
that's my long-term problem say telling. But in the short term, right now, what I worry most is actually dengue. Even
Okay.
if you have seen today's news, there are like 14 people died in because of dengue, right?
Okay.
So I know dengue for almost 2 decades now. Okay,
Mm-hmm.
and in Bangladesh for more than 2 decades, um, Bangladesh, all governments so far, every government so far, miserably failed to prevent this, the mosquitoes carrying the dengue virus, right? This
Okay.
is such a big failure, right? So,
Yeah.
if you give me a couple of minutes, I just want to talk about a little more
Yeah.
because
Yeah. Sure. Sure.
yeah, this is right now. I think we need a different strategy to control dengue. I don't believe anymore that Bangladesh government would ever be able to control mosquitoes.
I
I
think
think it is time.
the body that work to prevent this kind of diseases in Bangladesh is IDCR.
Yes.
So they should be careful about this. And
No, IDCR is the authority to do surveillance.
Mhm.
So they're like CDC, say for example, right?
Yeah. Yeah.
So, but it's up to the
Government.
executive part of the government,
Okay.
right? Who would implement. Definitely, IDCR has their role as well.
Mm.
IDCR need to bring the data to the government, and IDCR need to show the path what
Yeah.
needed to be done,
And
right?
push for efforts. Yeah.
Exactly, exactly. So, but there's a new development in dengue now. Like, I mean, I couldn't tell it like, if you would have asked me this same question 5 years ago, but I
Mm.
can tell it now
Okay.
because there is a new development of some really effective vaccines. So the first vaccine that was FDA approved is called dengue vaccine, which actually did not perform very well. So there are some side effect, and so eventually, US CDC and FDA limited the use of that vaccine to certain age group only,
Hmm.
and the patients, who probably had a previous history of dengue. But the dengue is not a problem in North America because it's not endemic. The dengue that happens here extremely scarce, extremely rare, and a few cases that happens here are because of the travel, right?
Yeah.
So going back to Bangladesh, India, Nepal, Sri Lanka, or Malaysia. So
It is considered that travel vaccine or something.
yeah, but the new developments are like there is a new vaccine is called TAC 003, and
Mm.
which also has been approved in some countries, which is
Okay.
showing a lot more promise. And the latest one is TB 005, which is actually developed by NIH USA. And an encouraging thing is that ICTD already did a clinical trial
Hmm.
um, on not on the effectiveness. They have done the safety trial.
Okay.
So the safety trial is done. This is the prerequisite before you go to the actual phase 3 clinical trial.
Okay. Okay.
So, but this publication came in already 2 years, and I haven't so far seen any new initiative to actually do the third phase 3 clinical trial. It's
Okay.
really, really urgent for Bangladesh right now to do this trial in collaboration to WHO as well as Gavi. Gavi is the
Yeah. Yeah.
Global Alliance for Vaccine, Global Vaccine Alliance,
And
and
the
then
biggest
they also
funding
actually
agency.
provide
Yeah.
a lot of support to the developing countries
Yeah.
for vaccine rollout. So in Bangladesh, I mean, actually, my heart breaks often. I often see in Facebook, among see that apparently healthy child, 5 years old, just
Mm.
abruptly died because of dengue. So this is a completely preventable disease. I mean, if you cannot prevent, contain the mosquitoes, I think it is time to start planning for vaccination. So yeah,
That's
so
true. Yeah,
I
yeah,
think the government really should take an active initiative in this regard.
yeah,
I
yeah.
mean, unfortunately, in our country, unless thousands of people dies, nobody notices this thing. But every day, like on an average, 10 to 20 people are dying. And if you
Mm.
just add up how many people are dying for a completely preventable case, it is very unfortunate to think that.
Yeah, yeah. Definitely, our government should focus more on that part. Yeah, thank you for sharing this information. So now let me focus on a little bit about your current research. So what are some of the projects that you are working on your current research lab as a part of the research or clinical labs?
Yeah, so my main research area for the last, I'll say, 12 years, right? Almost a decade,
Hmm.
is to apply novel technologies, molecular techniques on infectious disease diagnosis and surveillance. So
Hmm.
in particular, I already mentioned to you, I have developed nearly 60 infectious disease tests, which are based on mainly qPCR. This test has been implemented in three hospitals where I worked, right? In starting
That's
from BC
amazing.
Children's
Yeah.
Hospital in Qatar, and also now here. But at the same time, one of the main research area I have been working on is called metagenomics, clinical
Okay.
metagenomics. So just to give you an example to make it for the audience, to
Yeah, yeah,
explain it in
sure.
an easy way, I compare it with like when you go to fishing, you can use a fishing rod and catch one fish at a time, right? So that compare you can compare that with PCR. With PCR assay, we target a pathogen and we
Hmm.
only detect that pathogen
So,
only. We
yeah,
don't know
amplify that particular gene and then yeah.
exactly. So if the sample has any other pathogen, we would
Hmm.
not know by PCR. We would
Yeah.
only know, yeah. So, but PCR is really, really a good test for microbiology because it is really faster. Like, the
Hmm.
turnaround time, significant improvement, and high sensitivity, high specificity. But how many PCR you can have? There are hundreds to thousands of pathogens, right? So one diagnostic lab can afford to have maybe 20, 30, or 50 PCR assays
Yeah.
maximum.
I saw sometimes people use Techman PCR to
Yes, yes.
identify multiple, maybe. Yeah.
Yeah, so even with the panel, they are called syndromic panel.
Hmm.
So
Yeah, yeah.
even with panel, we also run many panels. We have respiratory panel, we have
Yeah.
gastrointestinal
If there
panel.
is something not targeted, not we will
Yeah,
not be able to identify. Yeah.
exactly. So even if we do a PCR panel, say for example, we can do maximum 15 to 20
Right.
pathogens at a time, right?
Exactly.
But the number of pathogens that can cause infection, especially this is particularly true for patients who have weak or compromised immune system, because a lot of bacteria, virus, or fungus, say for example,
Hmm.
they do not cause disease or they do not cause infection in apparently healthy body, but the same bacteria, to you it is like a part of your normal flora. The same bacteria can cause a severe life threatening disease in a patient who is vulnerable, in an ICU patient, in a cancer patient, right? So that's why we started to thinking, how do we apply next generation sequencing in
Hmm.
infectious disease diagnostics? I mean, one major challenge was that most infectious disease tests are actually very inexpensive, but the next generation sequencing is quite expensive, right?
Yeah.
One sequencing run. But again, even if it is expensive, if you can save a life by the technique,
Hmm.
um, you would probably save a lot of hospital money, because number of days a patient in the hospital, number of drugs they take, right? So those all cost money.
Yeah.
So we started that. We started initially. I started these projects like 12 years ago in BC Children's Hospital. And then I continued this research in Qatar, and still now I'm continuing this. So
Okay.
we are continuously trying to improve our approach and improve
Hmm.
sensitivity and how we interpret results. So we are mainly focusing on particularly invasive type of infection, like like
Mm,
infection in the brain, you know, or surgical site infection or pneumonia, or endocarditis, like the infection in the heart, right? So all the life-threatening infections, those we target, and we also improve the approach by targeting so that we can reduce the cost and increase the sensitivity. So
okay.
that is one thing. So application of metagenomics for infectious disease diagnostics, in particular for vulnerable population. The other area I work is on antimicrobial resistance.
Mm,
So
another big sector. Yeah.
exactly, yeah. So here also I'm applying a targeted metagenomic tool to
Mm,
characterize or identify, instead of one, two, three, or four, five genes, I'm
mm,
trying to identify like hundreds of genes at the same time in the sample. So
okay.
and we are right now trying to develop the protocol and validate the protocol. And I actually just submitted a big CHR proposal to do a study, and I included Bangladesh in that project as well. If I'm lucky to get to be grant funded,
Mm,
we'll be able to actually test some patients from Dhaka Medical College Hospital as well. So, but even if this fund grant is not successful, I would continue, submitting to other potential sources as well. So
so
this is
for
what I
your,
would like to do is that right now,
I
yeah,
have a question. So for your AMR project, so are you
mhm.
trying to develop new antibiotics or what
No,
kind?
no. My research is not related to antibiotic discovery or anything, because
Mm,
I'm clinical microbiologist, right? So
okay.
we do clinical microbiology. My research is very closely related to the service we provide to the patient.
Mm,
So
okay.
new diagnostic assay development, or utilizing these assays
Mm,
to gather new knowledge on something. So
okay. Okay.
so right now, antibiotic screening methods are limited to,
And
like
yeah,
limited number of pathogens.
sometimes identifying the susceptibility and those things.
Yes, yes,
Sensitivity.
yes.
Okay.
Yeah. So, yeah, as well as the surveillance. So we also do whole genome sequencing of different
Mm,
viruses. We do whole genome sequencing of multi-drug resistant bacteria. We characterize them, and we see if there is anything unusual in those multi-drug resistance bacteria, right?
okay.
So that type of study also we do. Yeah. There are
Okay.
also many other different projects, but these are the main focus of my current research.
Okay. All right. I wish you all the best for your current research projects.
Thank you.
And thank you for sharing all those information and your insight on different infectious diseases. I think uh, it will be really helpful for our audience and everybody. And with that, we'll conclude our research talk. And in the next part, we'll talk about career, specifically how to be a licensed clinical microbiologist.